welcome back to Activate Albany. I'm Rachel, and today we're going to be doing a DNA extraction experiment using household ingredients and strawberries. So this is an experiment I actually learned in a course back in college, but you can actually recreate it with some stuff found around your pantry. Today you're going to need rubbing alcohol, a plastic bag, water, a strawberry, a little bit of salt, a skewer, a teaspoon, and dish soap, and if you can, 2 glasses, and a paper towel as well. So first you're going to start off with taking a strawberry and placing it right into a Ziploc baggie. And you're just going to close that right up and mash it. So if you're doing this with your kids at home, make sure you don't make too much of a mess for your parents. And once that's all mashed up, you can place that to the side while we prepare our buffer. So this is where things get interesting. In a glass, you're going to take a little bit of water and 1 squirt of dish soap. So dish soap actually breaks down the lipid bilayer of the cell membrane, which actually helps with the DNA extraction, and a little pinch of salt, or sodium chloride. And then you're just going to mix that all right up. And this is your buffer. You're going to take this and just pour it right into your bag with the strawberries. And then you're going to zip that right up, getting rid of all the air. And make sure you give it one more good mash. So while that is sitting to the side, you can prepare your second glass. And here, you're going to take a piece of paper towel and put it right over the top, leaving a little bit of a well. And then a rubber band, and you're going to secure that right to the top. After giving this a little more of a shake, I'm going to take this solution and pour it right into the well we placed in our cup. All right, so we placed a little bit in here, and while it starts to drain, we'll be right back in a few minutes. Alright, so it's been about 5 minutes since we've let our solution sit in the strainer. So while we've been waiting, the sodium has been removing the proteins from the DNA, and we're left with a clear liquid with a pinkish hue at the bottom of our cup. You don't need too much. We've got about, say, 1/4 of a teaspoon, but that's just fine. And now we're going to take some ethanol or rubbing alcohol. This is 70%. I've been letting it sit in the fridge for a couple minutes, so if it gets nice and cold, it's not required, but it does help remove the DNA protein. You don't need too much, but just enough to get enough so that some of the liquid in the bottom of your cup can float to the top. So I'm going to pour maybe 1/2 cup into the beaker right here. And I'm just going to wait a few more minutes and let the 2 solutions separate. So we're right back. It's been about 5-ish minutes. We've been letting our buffer solution sit, and the ethanol that basically rubbing alcohol helps the DNA precipitate and allows us to extract it from the solution. So what we're going to do is take a skewer. You can also use a chopstick, or even a very thin piece of metal if you have, or a paper clip. And you're going to take it and you're going to look for the little bit of stringy, you're going to see a little white stringy thing floating around. And that's your DNA. And that's it for today's episode of Activate Albany, and I'll see you guys next week.